Alright, so I thought I would go over some of these problems from Chapter 5. And for this little recording, we'll just do the ones that correspond to examples of problems involving one gene. So we've talked a lot about Mendelian genetics and how we can get various genotypic ratios and phenotypic ratios from standard Mendelian crosses. For example, a Mendelian mono-hybrid cross gives you a 1 to 2 to 1 genotypic ratio and a 3 to 1 phenotypic ratio. And a test cross would give you a 1 to 1 ratio. And so these problems are situations where we still get that type of inheritance in terms of how the genes are inherited. Like we might have a monohybrid cross and we'll get a 1 to 2 to 1 ratio. But the way we normally get from a 1 to 2 to 1 ratio to a 3 to 1 ratio is that the heterozygotes and those dominant homozygotes for the dominant allele both give us the dominant phenotype. So you get a 3 to 1. But there are variations on those themes where we see we'll get a 1 to 2 to 1 ratio, but we won't necessarily get a 3 to 1 ratio. And so this first example is one where we get that situation, but sometimes these are a little tricky because you have to look at this and figure out what the pattern is, and you're not necessarily given the pattern. And so in this case, we see palomino cross to palomino. We get 13 palomino, 6 chestnut, 5 cremello. The idea is that 13 is almost close to twice as much as this, and so this looks like 2 of these, 1 of these, and 1 of these, and so that looks like a 1 to 2 to 1 ratio. If it's a 1 to 2 to 1 ratio, then that means it's this, where we get 1 of these guys to 2 of these guys and 1 of these guys. And so our heterozygotes are the 2. So our heterozygotes are the palominos, and our heterozygotes are these. So that tells us that if we do a monohybrid cross, we get a 1 to 2 to 1 ratio, and that's characteristic of incomplete dominance and codominance. Because palomino is intermediate between chestnut and cremello, and there are pictures in the book that show you the pictures of these horses, and so that suggests incomplete dominance. So explain the inheritance pattern. Incomplete dominance. So that's basically what's going on. And then we can fill in the genotypes for all of these things. And let's get a little extra room here. So in this case, so what does that mean if it's incomplete dominance? Well, so the heterozygote gives us something in between 2 other things, but then these 2 other things are going to be the other 2 genotypes that are possible. So we could just in this case just make this up and say these are chestnut and these are cremello. By making it up, I just mean we can designate big A as chestnut, and then these are palomino. And then once we have that just tells us everything we need to know. So we'll say chestnut. We said that those were big A, and the only way to get it is this. And we get all chestnut, and that makes sense. Cremello, we said that was little a, and we get all cremello, little a. Palomino, they have to be heterozygous. So the idea is that you figure out these rules, you figure out what's going on, and once you have that, it's easy to go through and say what everything else is. And so we said chestnut was big A, and so if this is like a test cross, then we would have to get 1/2 would be this, and 1/2 would be this, and that makes sense. This looks like another test cross, big A, little a, cross to the cremello genotype, 1 to 1 this. And then chestnut cross to cremello will give us all palomino, and that makes sense too. So the tricky part there is just sort of getting started and identifying that 1 to 2 to 1 ratio, and then everything else sort of falls into place. Here, the information is given to us ahead of time, so that's useful. It says that M and N alleles are blood groups that exhibit codominance. So that if it's codominance, that means this gives you the M phenotype. This gives you the N phenotype. And this is sort of like blood groups, like the ABO blood groups that you're more familiar with. It's just another example of something where you get a protein or some other antigen expressed on the surface of a cell, of a blood cell. And you can have two different types of antigens on the surface. You can have, have either M, or you can have N, or you can have M and N. And then if you just test for the presence of these, if you have a heterozygote, you'll get both of them. So both the M allele and the N allele are expressed, so that makes them heterozygotes. I mean, that makes them codominant. So give the expected genotypes and phenotypes in these progeny. So this is, so they're already giving us the genotypes of these. So what we would get here is we would get M, or M, N. So those are the genotypes. And then the phenotypes are just M blood type or M, N blood type. For these guys, we could only get the N genotype, the N genotype, and only the N phenotype. These guys, this is this one here is it looks like a monohybrid cross. So let's get a little more room. And a monohybrid cross gives you, we would get 1 MM to 2 MN to 1 NN. Those are the genotypes. And then the phenotypes would be M, MN, and N. Here we have a test cross, a heterozygote cross to double heterozygote. Well, I guess based on my previous definition of a test cross, where this would always have to be homozygous recessive, it's not technically a test cross because we're not seeing, these are codominant, so it's not that one is recessive to the other. But it should still give us one of this genotype, one of this genotype, and then the phenotypes are M or N. So that's all that's saying. Here we have long earlobes. This is just to illustrate the concept of penetrance. So we have long earlobes is autosomal dominant. So L equals long, little l equals short, and we've got 30% penetrance. So all this means is that if you have a particular genotype, there's only a 30% chance that you'll actually exhibit the phenotype. So all of these are sort of a difference, or sort of a mismatch between what the genotype is and what the phenotype is. And so here we have a person who's heterozygous for long ears, and maybe with a person who's homozygous for normal ears. What's the probability that their first child will have long earlobes? Well, we've got a 1/2 probability of getting a big L, because we can get either that allele or that allele. That's segregation of alleles. We have to get a little l from this parent. So there's a 1/2 probability of getting the genotype, but there's only a 30% chance of getting the phenotype. So let's put that in percent. So 1/2 of 30 is 15. So you have a 15% chance of getting the first child having long earlobes. Here's an example we've got your Chinese hamsters. And we've got a Chinese hamster with white spots. It's crossed with another hamster that has no spots. So just at the phenotypic level, we're going to go, we'll just call it spots crossed to no spots. And we get a one half with spots and one half with no spots. So that looks like a test cross of a monohybrid. We get a 1-to-1. And so one of these is heterozygous, and one of them is homozygous recessive. But we don't know which one is which. So we don't know. Let's say we just don't know yet, because it could be either way. So we could have if spots is heterozygous and no spots is homozygous recessive, we get 1-to-1. If no spots is heterozygous and spots is homozygous recessive, we get 1-to-1. So we don't quite know yet. So with two hamsters with white spots are crossed, so now we go spots cross the spots. Now we get 2/3 with spots and 1/3 without spots. So now this is where we can see that this is a modified version of a 1-to-2-to-1 ratio. So it's a 1-to-2-to-1 ratio. We're just missing one of the ones. And a 1-to-2-to-1 ratio is this. And so we're seeing that the 2 is giving us spots. So that means spots is dominant. So if we can say we get this, and we get spots, and we get little a will give us no spots. All right. And so what's going on there is anytime you see a 2/3 to 1/3, that means that we're not seeing, we're not seeing this. And so we interpret that as those guys dying. We do see this. These guys have spots. And we do see this. These guys have no spots. So big A, or whatever you want to call it, is dominant to little a, but it's recessive lethal. You could call it homozygous lethal, and then you could not worry about the term recessive. But when homozygous, they die. When heterozygous, you get spots. When homozygous for the other allele, you get no spots. How about you go about producing Chinese hamsters that breed true for white spotting? You can't, because you can't. This would be true breeding. You can't get that because these guys are both dead. There may be some ways that you could, if you get these guys for spotting and you wanted to make those true breeding, I just don't think there's a way to do it, because these guys will never be true breeding because they're heterozygotes. You'll always get 1/3 of these will come out as having no spots. So it's kind of a trick question. Can't do it. All right, and so here we have to pause. All right, so here is number 18, and this is a series of chi-square problems, or sort of different chi-square. So this is just using some real data to look at some chi-square examples, and there's these series of experiments. And they were looking at yellow coat in mice, and as we know from the chapter, that it actually turns out to be a lethal allele that shows a 2-to-1 ratio. But when they first did the experiment, they looked at it and thought it looked like a 3-to-1 ratio. And so they look at these numbers, and so we're going to do chi-square on the various numbers. So we're going to look at however you pronounce that name, Q-no, I don't know, or capital and little, or both combined. So rather than going through all the math slowly, here I just copied the math from the solutions manual and put that in here, and that shows us how to go through chi-square again. And so we have the observed over here. It's important to add up the observed, so we get a total. And then we have to calculate the expected. So first they did it based on a 3-to-1 ratio. So where do they get these numbers? They take 363 times 3/4 to get 3/4 expected here, and times 1/4 to get this expected here. Then you subtract these, and then square them and divide them by expected, and you come up with a chi-square value. So this is a sum of chi-squared, because it's chi-squared for each of these things added up. We have 1 degree of freedom because we only have 2 categories. And then we get 1.257. Then we go to a chi-square table, and here let's just take part of the, and let's put it, here we'll just pop it over to a new document over here. So it's just a little chi-square table. And then we can go back and forth between our problems in the chi-square table. So we get a chi-square of 1.257. So chi-square so we had a chi-square of 1.257, and we look at this, and so chi-square of 1.257, 1 degree of freedom, 1.257 comes in here somewhere. So our p-value is greater than 0.05. That means the probability that the difference between observed and expected is due to chance is relatively high. So this is, what is the probability that O equals E? That's the way I like to think about it now. The probability is between 0.1 and 0.9. So that's a high probability that O equals E. So we do not reject. Okay. So we cannot reject the hypothesis of a 3-to-1 ratio. Because our p-value ended up being greater than 0.05. So when they did it for a 2-to-1 ratio, they got a chi-square value of 5.46. If we go to the chi-square table, oops, sorry, I think it was chi-square was like 5, so 5.46. So here's the same data, chi-square, and this was testing the 3-to-1 model. This is testing the 2-to-1 model. So 5.46, that's a higher chi-square value, so it's way down here, 1 degree of freedom. That comes in here somewhere. So that in that case, the p is less than 0.05, so we have to reject that hypothesis. Turns out that it really is a 2-to-1 ratio, but just based on these data, the p was less than 0.05, so we reject the 2-to-1 hypothesis. So just based on that data, they thought it was a 3-to-1 ratio. They were wrong. Sometimes people are wrong. And so then let's look at then when they redid the experiment, or when these other dudes redid the experiment, they got some different numbers. So these are the Castle and Little results. And they counted up 1,000 mice. It's a lot of mice. And got 800 yellow, 435 not yellow. Here they're testing the 3-to-1 ratio. So they're calculating their expected based on 3-to-1. So they multiply the total by 3/4, and they get this. Multiply the total by 1/4, and they get this. They do a chi-square, and they get a huge chi-square. So that's a huge chi-square, and with 1 degree of freedom, that's going to be way to the right, and you can get a really, really low. So if you get really high chi-square, chi-square goes up, p-value goes down. So you're going to have a really, really low p-value. And I'll let you go back to the table, but it's less than 0.005. So we really reject this one at a very low p-value. This one, however, if you test a 2-to-1 ratio in this case, you get a chi-square value of 1.98. Look it up in the chart. Let's go look it up, 1.98. 1 degree of freedom. 1.98 is somewhere in here. That's greater than 0.05. So p-value we do not reject. Example involving just one gene, this red pearl millet plant, whatever that is. Color is determined by 3 alleles. So RP1, this would just go away. RP1 is red. RP2 is purple, and just little RP is green. So RP1 is dominant to, so this gives us this little info here. RP1 is dominant to RP2, RP2 is dominant to little rp, and RP1 is also dominant to little rp. So that's what we call an allelic series. An allele means different versions of the same gene, so we're just talking about one gene, 3 different potential alleles, but only 2 in any given organism. So in this case, RP1 over RP2, RP1 is dominant to RP2, RP1 is red, so we get red. RP1 is also dominant to the other one, so we get red. And then we'll get RP1, 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 little rp, RP1, RP2, and RP2, RP1. So that's just saying we get this combination, this combination, this combination, and this combination. So we're just getting all 4 possible combinations from those things. Anything that gets an RP1 is red, and then this one should be RP2, and then little rp. But anything that gets an RP1, so we can get RP1 from the first parent or an RP1 from the second parent. If we get any of those, we're red, so we get 3 red. And then RP2 and RP, that's, let's see, RP2 is purple, so we get 1 purple. So we get a 3-to-1, which is consistent with one gene, and we got, these things are heterozygous for 2 things. This thing, we can get, let's just go with 1 and 2 together, 1 and RP together, 2 and RP together, and then RP and RP together. This is the only way to get green. 1 and 2 is red. And then we get 1, it's red. 2 and RP, that's purple. And so I guess I should put this in here. RP1, RP1, RP2, and then this is RP2. So really we get 2 reds, 1 purple, and 1 green. So 2 of these, 1 of these, and 1 of these. So that's a 1 to 2 to 1 ratio. So that's, you know, again, we'll see 1 to 2 to 1, and it just, and up here we saw 3 to 1. When we start seeing 1 to 2 to 1 and 3 to 1, that suggests that 2 genes are involved. I'm sorry, 1 gene is involved. This one, RP1, RP2, RP1, RP2, that's 2 heterozygotes crossed together, so it's got to be 1 to 2 to 1, something to something. RP1 and RP1, that's going to be red. RP1 and RP2, it's going to be red. RP2 and RP1 is going to be red, so it'll be two heterozygotes that are red. Then we can get RP2 and RP2, and those are going to be purple. So here we'll get 3 to 1 red to purple. So we had RP1 from the first parent, RP1 from the second parent. RP1 from the first parent, RP1, 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 RP2, and then we could also get RP2 from the first parent and RP1 from the second parent. And this is our two ways of getting heterozygotes, our one way of getting that homozygote, and then we can also get RP2, RP2, and there's only one way to get that. Two ways to get that, one way to get that, 3 to 1. This looks more like a test cross, so we can get RP2 and RP, or we can get RP and RP. These guys will be purple. These guys will be green. It's a 1-to-1 ratio. And these guys were purple. These guys were green. Same thing down here. It's kind of a test cross going the other way. So we can get RP1, little rp, RP2, little rp. These guys are red. These guys are purple. So what we're seeing is various combinations of 1-to-1, 1 to 2 to 1, 3 to 1. When you see that, you got one gene involved. Either monohybrid crosses, like these guys, or test crosses, like these guys. Alright, so then all these other ones will be pretty much the same. So if we look at B, you can, now we got RP1. Well, first of all, we just put in all the parents. That makes life easy. So RP1 is dominant, so that's red. RP2 is dominant, so that's purple. RP1 is dominant, that's red. RP1 is dominant, that's red. RP2 is dominant, that's purple. Oh, there's a green one. So the only way to get green is if you get these two little RP alleles, because those are recessive. And these guys are green. RP1 is dominant, that's red. So for B, we get RP1 over RP. Goes to RP2 over little rp. So now we can get RP1 over RP2. That's one of the results we can get. So this would, we're going to get 1 to 2 to 1 to 2 to 1 type thing. We can get this and this. We can get this and this. We can get this and this, and we can get this and this. Do Punnett square if you want. So we got RP1 with RP2. We can get RP1 with little rp. We can get little rp from that parent with RP2. We can get little rp, little rp. These are red. These are red. That's not a 1. That's a little rp over rp2. rp2 is purple. And little rp is green. So we're going to get one of these, and 2 of these. And so that's a 1 to 2 to 1 ratio. That's indicative of one gene in some kind of a monohybrid cross. This one up here was a 3 to 1, which indicates one gene. So think monohybrid cross. And I'll leave you to go look at the answers in the solutions manual because I'm running out of room here. Yeah, we'll do one more. And then I think you get the idea. Man, sorry about that. All right, so over here we got C. RP1 over RP2 cross to RP1 over RP2. So that's straight up dihybrid cross here, because we got heterozygous for the same things. So we know there's one, we can get RP1, RP1, get one of those. We can get RP1, RP2, so we get one of those, two of these, and we can get RP2, RP2, we can get one of these. These are red, these are red, and these are purple. 3 to 1. Because this is just a monohybrid cross. So, and RP1 is dominant over RP2. So we'll get standard monohybrid cross stuff coming up. So that's all good. Same thing here. We've got some rabbits. We've got an allelic series. There's nothing too crazy going on. Need a little room to write our answers so we don't run into the same problem we just did. Alright, so we've got our allelic series right here. And we just have to figure out what's going on. And so what you do with these is you write in what you know, and then you put a blank where something you don't know, and then you fill in the rest. So we know full color has to have a big C, but it could have anything else because it's dominant to everything. Albino is recessive, so it gets two little C's, and then we get some full colors out, and we get some albinos out. Well, the only way to get albinos out is if we get a little C from both parents, so that means we have to have a little C for that parent. And then that means we'd end up having a little C there. Himalayan is CH, but in order to be Himalayan, it could have two CHs, or it could have a CH and then a little C. So we don't know yet. But these recessive guys are nice, right? So we can do a test cross, and then we can fill out what's in here. Then we get one to one Himalayan to albino, so that means we must have had so we're getting some albinos out, so we had to have a little C here. And then that makes sense, because then we could get 1 to 1 Himalayan to albino. So it's pretty easy if you do a test cross, because that really sorts things out. This one, full color, has to have a big C, little C, or this could be anything. Then out of here we're getting full color. So that means we got a big C, but here we're getting chinchilla, so that means we got C little CH. And so we have to get a little C from this parent in both cases, but this CCH is dominant to the little c, so that gives us chinchilla, and that had to be coming from here. Here we've got big C blank across to Himalayan, which we know has to have the Himalayan allele and something else. Then we get half full color, half full color. Then we get some Himalayans out. So Himalayans, we got to have a Himalayan allele. And albino, we have to have two little C's. So, and this is a 1 to 2 to 1 ratio. So this is the 2, and then we get one of these and one of these. And so, in order to get albino, we had to have a little c here and a little c here. And then we'll get half of the offspring will get this big C, and then either this or this. So these guys could be C, CH, or big C, little c, and those would both be full color. You could get this CH allele from this parent, and then a little c from the other parent, and then that would be Himalayan, or you could get two little c's. Fill that up. One more, full color, across to full color. Then we see a 3 to 1 of full color to albino. That means if we get 3 to 1 and we're getting albino out, that's just a typical monohybrid cross. We get 1 CC, 2 big C little c, and 1 little c little c. And then these guys are both full, and these are albino, 3 to 1. So they're just monohybrid crosses. But we've got different allelic relationships. Here this is starting to show a little bit of, this is blood type, and it's looking at blood type at 2 different loci. And so we're looking at, the mom has blood type A, so that means she has an A allele, and then either an A allele or an O allele. And she also has M blood, which if she has M blood, she has to have an MM genotype, because that's, in that case, there's only 2 alleles. There's big M and big N, and if you have M and N, you get MN blood type. So she has to have that. The child has A, B, M, N. I think the best way to figure this out is that they had to get the B and the N from a parent, from the father. So George doesn't have a B, so it can't be him. Tom has a B and an N, so it could be him. Bill has a B and an N, so it could be him. Claude does not have a B, so it can't be him. Henry has a B but no N, so it can't be him. So it's got to be one of these 2. So you can't rule them out. Alright, so I'm going to add a problem here. Starting at 25, we start looking at 2 genes and some gene interaction like epistasis and that sort of thing. So we'll hold off on that till later. But for now, I do want to add one more type of problem here, just to show that now that we've got different types of inheritance, so let's just try a new problem that we'll just make up on the spot here. So let's say we have a yellow mouse. And it's the same yellow mouse that we had before, where the yellow allele is lethal, and we know that the yellow is a lethal allele. And we cross that, and this mouse also has a short tail. And short tail is just recessive. Short tail, long tail. And little y equals a brown mouse. And y is yellow, and it's dominant to brown, but it's also a lethal allele. So we're going to take a yellow mouse with a short tail, cross it to a yellow mouse with a long tail, and see what we get. Alright, so we know that the yellow mouse is heterozygous, because big Y is dominant to little y, and if you have 2 big Y's, you're dead. So if you're a yellow mouse, you have to have 2 big Y's. So that's a big Y and a little y. And then we can have 2 little t's for that parent. And the other parent has to be big Y, little y. And there's a long tail, so it could have a big T, and it could have a little t or a big T, but we're going to give it a big T for this problem. So what could this give us? Well, this guy could give us, it could give us 2/3, so it's going to give us some of these. It's going to give us 2/3 yellow, 1/3 brown, and all of these will have long tails. Stop it. So now the question is, what if we cross two of those together? But let's take the yellow ones. We're going to take two of these and cross them together. All right, so what does that look like? Well, that looks like a dihybrid cross. So but we can't do 9331 because 9331 depends on each of these things behaving normally by itself. But we know that the yellow gene is going to give us 2/3 yellow and 1/3 brown. And so that'll screw up a 9-3 to 3-1 cross. 9-3 to 3-1 depends on this being 3/4 and this being 3/4 or 1/4. Now we also know that, but then for tall, or I mean not tall, but for the long tail, we expect a long tail. What, 3/4 long tail, 1/4 short tail. And then we can easily predict what we would get in the F2, or get in the offspring of this particular cross. Now it's important that we took the yellow guys here and crossed them, because if we took these would be homozygous for two little yellows, and then everything would be brown, and we would just have 3/4 long and 1/4 short. So we took this 2/3 yellow so that we could look what happens when we look at these two different types of ratios and combine those. So now we're just combining probabilities. So how many are going to be yellow and long? 2/3 times 3/4 is 6/12, and that's yellow and long. Two-thirds yellow, 1/4 short is 2/12 yellow and short. And then 1/3 brown across times 3/4 long is 3/12 brown and long. And then we'll get 1/12 brown and short. 6-2 to 3-1. Now that's not something that you should memorize necessarily. It's just you figure it out. You know, we could put a third gene here, some eye color thing maybe, and then we can figure out how many of them. We'd have 3/4, 1/4 for this gene. So you don't need to memorize all the different ratios. You just figure out what's going on with one gene, figure out what's going on with the other gene, and multiply those together. And you can do that as long as these genes sort independently. So that's a type of problem where the whole point here is to have a weird ratio for one thing, have a different ratio for the other thing, and just multiply those ratios together to see what you get when you do the crosses at the same time together. All right, so that's it for this part of Chapter 5, and we'll move on to the second half of Chapter 5 problems after the first exam, so that we can look at what happens when we've got two genes that are actually interacting with each other. So these genes aren't interacting with each other. These are two completely different traits, and we're just looking at the inheritance of both those traits at the same time. Next time we'll start to look at two different genes that may or may not assort independently, but they both contribute to the same trait. So we start to see different ratios in that sense.